Okay, hi everyone. Before we start today, I wanted to remind those of you who have Friday recitation that this week, because of Kuhio Day, it's a school holiday. But I spoke with the Friday TIs during our weekly meeting, and we came up with the idea, or actually they came up with the idea that your TIs for the Friday sections are going to make recordings of their review of the lecture material for you to watch. So I strongly encourage you, to make use of that, right? To take advantage of the fact that your TIs are making these nice videos for you to help you review the material that we've been learning about this week. For the entire class, so obviously for those of you who have Thursday recitation sections, you'll have normal recitation, right? Because Thursday is still a normal school day. Okay. But for the entire class, there is no quiz this week because we just finished with the exam and then had spring break. But there will be a new homework assignment that is assigned starting this week. So all the more reason to make sure that you attend recitation or watch your TI's recitation video so that you are set up really well to understand the material and you can start doing your homework that's due next week. Okay. So let's talk about enzymes. What are enzymes? I think we all, this is one of those things, again, where we all have some sort of intuitive notion of enzymes because we, heard about enzymes. Those of you who maybe wear contact lenses, maybe you're aware that certain types of, enzymes in your multi-purpose contact lens solution. But what are enzymes, right? Why are they so important biologically, and why do they play such a prominent role in our living cells? So let's start off with by kind of revisiting this notion of energy, right? Because as we discussed last time, the biochemical reactions that take place inside of the living cells inside of your body that allow you and I to be alive and function, right, are all due to reactions between biological molecules having to do with energy, right? The energy that's contained or represented in chemical bonds. So the electrons that are in orbits, right, as atoms are either sharing their electrons in shared orbits with each other, like covalent bonds, or if you just have atoms that are sort of close to each other because of attractions due to different charges. Right? It's all about the energy that's represented by those electrons moving around in those electron clouds, right, in those orbits. Remember last time we said that a fundamental sort of simple way of thinking about the definition of energy is simply energy is the ability of matter, so atoms, right, because atoms make up matter, to move. Right? And it's also can be thought of as either the potential amount of energy that's stored in electrons moving around in their orbitals, right, or kinetic energy when those electrons are sort of liberated from those bonds, right, and momentarily, right, are seeking out new bonds to re-engage in, right? And that can be kinetic energy, the release of some excess energy that might be able to be harnessed by other bonds of other molecules nearby, right? If things are set up properly, right? So you actually have different molecules at the right spot at the right time in order to take advantage of certain bonds breaking, right? And that energy being used to encourage other bonds in other molecules to also undergo change, right? So that's what we talk about when we talk about energy at the atomic level. How do we measure energy? We last time already mentioned the term kilocalories. We looked at that as an example of when we're, for example, looking at how much potential energy is represented in all of the atomic bonds of something like one glucose molecule, right? All the carbon-carbon bonds in there. I mean, those are certainly electrons that are in orbits, right? And those certain amount of energy in those bonds. We said that if you were to essentially liberate the amount of energy in those bonds, right, you'd be able to free up something like 686 kcals of energy from the metabolic breakdown of one glucose molecule, right? One C6H12O6 being broken down eventually through numerous electron orbital interactions changing, bonds breaking and reforming, down to something like carbon dioxide and water, right? And that would release 686 kcals. So what is a kcal? What is a calorie? So 1 calorie, in this case with a lowercase c, is the amount of energy needed to heat 1 gram of water, H2O, 1 degree Celsius at normal, sea level atmospheric pressure of 1 atm. Right, so that's 1 lowercase c, the amount of energy that's needed to heat 1 gram of water, 1 degree Celsius. Now, you and I usually don't think about calories in that sense, because that might seem like a teeny tiny amount of energy. We typically think of calories when we're thinking about dietary considerations, right? Like food labels. In that case, 1 calorie is a capital C calorie, and that represents 1,000 lowercase c calories. Right? So 1 calorie with a capital C is 1 kcal, right? 1,000 lowercase c calories. All right, great. So that's one way of measuring the amount of energy. But let's think about this in terms of energy and the motion of bonds and the ability of bonds to break and reform based upon energy. So here's an ice cube, and on the right is the molecular structure of the water molecules in that crystal of ice. Right, so here we have a whole bunch of H2Os. So the reds are the oxygens and the 2 little white spheres are the hydrogens. And if there is not much energy in this environment, right, that means there's not a lot of potential for these molecules to be vibrating around or moving around, right? Then if the energy is low enough, then the attractions of the hydrogen bonds of the water molecules, right, is stronger than any kind of vibrations of the water molecules themselves due to the amount of energy that's surrounding there. So at 0 degrees Celsius or 32 degrees Fahrenheit, right, the energy of the environment is low enough that the water molecules stop vibrating around to the point where the interactions between the hydrogen bonds are actually strong enough to hold these molecules together in a very orderly, repeating crystal pattern, right? Which is what you see here. And by the way, you notice that part of this pattern is that when water molecules freeze or come together because of their hydrogen bonds being stronger than any other vibrations of these water molecules, right? They tend to form these little hexagonal or 6-sided structures. Right? And that's why that as an ice crystal grows, right, and gets larger and larger and larger, as more water molecules sort of, chill out, pun intended, right, and are able to actually form contributing hydrogen bonds, the growing crystal is 6-sided, right? And that's why snowflakes tend to be 6-sided, right? Because they're just big representations of the fact that the crystals that are growing have 6 sides to grow from as more water molecules get added to them. Right? So this is actually kind of an entertaining animation here. What happens to the water molecules in this crystal structure once you start heating things up? Right? So if this image is at 0 degrees Celsius, what happens when you start putting more energy into this in the form of, for example, heat? Right? Heat is energy that will cause the water molecules to start vibrating more and more and more. And at a certain point, what? The vibrations are intense enough that they are enough to disrupt the very weak hydrogen bonds. Right? And what do you think happens? Well, the water molecules fall out of this crystal structure, right? And that's how you end up with liquid water. So let's see this animation. So it starts off at 0 degrees Celsius. Here's the crystal structure ice. Oops, sorry. I meant to start this thing up here. As you start putting more energy into it, right, the water molecules vibrate, because that's energy, is the motion of matter, right? It gets to the point where that amount of vibration, right, is enough to disrupt the weak hydrogen bonding between the water molecules, and now you have liquid water, right? If you cool this down, right, and start allowing the water molecules to kind of stop vibrating so violently, then they will naturally start re-engaging with their hydrogen bonding, and that's how the water freezes again as ice. Right? Now, the far extreme would be if you heat this up to 100 degrees Celsius, what will happen? There's so much motion, so much vibration of the water molecules that then the water molecules begin to escape, right, from the liquid into the surrounding air. And that's when you start boiling the water and you have water vapor, right, or steam. Okay, so I want you to keep that in mind, that when we're talking about energy, that's what we're talking about, right? How much energy is needed to get atoms to shake around enough that it overcomes whatever bonds they have holding them together as part of a molecule. For water, we're talking about the weak hydrogen bonding network, right? That's really weak, right? Because it's just between the H's and the H's of one water molecule and maybe the oxygen of another water molecule kind of interacting together. Right? But for more complex molecules, we're talking about things like the covalent bonds that are actually taking place inside of the molecule between things like carbons and hydrogens or carbons and oxygens, right? But the principle is still the same. So let's think about this idea of calories again, right? So that's how we measure the amount of energy that we're dealing with in a chemical reaction. So if we talk about our sort of normal everyday, familiarity with calories with a capital C, we can look at things like nutrition labels. So here's Funyuns, right? Funyuns and Coke. That's what I pretty much survived on when I was in your guys' shoes as a college student. Funyuns, Coke, and the occasional pizza, right? But let's look at this. So the nutritional label says 140 calories per 13 pieces of Funyuns, right? And so that gives you 140 calories with a capital C. So that means 140 kilocalories, or 140,000 individual calories. So if you were to actually convert all of these Funyuns in this bag, or 13 pieces of Funyuns, right, into, energy, that implies that you'd have enough energy in a bag of Funyuns to heat, 140,000 grams of water, or 140,000 mLs of water, 1 degree Celsius, right? So that's actually quite a bit of energy if you think about it. Let's look at Coke. Right, here's a bottle of regular Coke. Coke has 240 calories, right, in one can, right, with almost all of it just coming from sugar. So let's have some fun with this, with the Coke label here. Can you actually gain anything useful, knowledge-wise, about the energy contained in a glucose molecule just by looking at the Coke label? So let's have a fun exercise. The Coke label says there are 240 calories with a capital C if you drink this one can of Coke, right? This, in this case, this is a 20 fluid ounce, maybe bottle of Coke, right? And that's all contained in 65 grams of sugar. So could you use this to actually estimate how many kcals of energy are there per, let's say, a mole of glucose? So 1 mole of glucose is 6.02 times 10 to the 23 molecules of glucose. And 1 mole of glucose would weigh 180 grams, right? That you could just figure out by looking at the periodic table and calculating the molecular weight of carbon, hydrogens, oxygens that make up, a glucose molecule. So I should mention that in this bottle of Coke here, the label says that the sugar is in the form of fructose, right? So fructose and glucose are the same chemical structure, same chemical formula, just slightly different arrangement, right? So let's use this. So if you say, well, you could do this calculation. If you have, how many calories, kcals are you talking about here per grams of sugar, right? Times 180 grams per 1 mole of sugar, you can then estimate how many kcals per mole of energy there is contained in glucose, right? So we can just punch that in. I have my calculator here. So calories, so 240 kcals based upon this label here, divided by 65 grams of sugar are giving you those 240 kcals, times 180 grams of sugar per mole of sugar. So that gives you, I don't know if you can see that, right? So that gives you 665 kcals per mole of energy that's contained in glucose. And how accurate is that? I mean, that is pretty accurate, right? Because last lecture we just said that the energy content of glucose is 686 kcals per mole, right? So that's pretty close. So it's kind of cool that you can actually do some biochemical calculations just based upon the label that you find in the supermarket. Okay. So last time we introduced this concept of free energy, and it's a way of thinking about all the atomic bonds, the molecular bonds that are in a biological system. And I said a system, it's up to you to define what a system is. It could be an individual living cell is a system, right? Or it could be your entire body. You know, you are one unit of a human being, right? You could say that's one living system. Right? In either case, you're talking about, you know, a multitude of different molecules, each with different bonds, right, as part of their structures, and each of those bonds represents electrons that are in some energy state, high or low, that are available, right, to be broken and interact with other at the formation of other bonds, and therefore the change in energy in the system is very dynamic, right? Some interactions make things go up, some make things go down in terms of energy. Recall that the change in free energy, or delta G, is positive if you're making more complicated, elaborate molecules from simpler ones, right? Because you're taking maybe electrons or chemical bonds that are in lower energy states in simpler molecules, right? You're breaking them and actually putting them together in more complicated structures. So the free energy represented by that bigger, more complicated molecule is in the plus delta G direction. Right? Inversely, if you took a complex molecule and started breaking all those bonds, right, to kind of liberate that energy, that potential energy that's in there, and you end up breaking it down, interacting all the atoms in different ways to get simpler molecules, that is a negative delta G direction, right? Because there'd be some energy that's released. Right? So that negative delta G direction is what we call the thermodynamically spontaneous direction of reactions. Right? Spontaneous in this case, we have to be careful about this term. Spontaneous does not mean that things will happen by themselves, right? We did look at an example of extremes. If you have a very strong oxidizing reagent and a very strong reducing reagent, for example, potassium permanganate and glycerol, right? One is a very strong oxidizing reagent, one is a strong reducing agent. That means if you mix them together, hey, there's a real strong likelihood that electrons are going to be exchanged, right? And new bonds are going to be formed. And that's why you have a very strong high exothermic reaction, right? You mix those two things together and you're going to get flame, right? The release of heat, as all of the potential energy in those electrons in the more complicated molecules, right, are then broken and reforming different compounds, and all of that energy is just released at once. Right? So we said that that reaction might happen spontaneously, right? And certainly that's a thermodynamically spontaneous direction that it goes in, right? But not all biological molecules, and in fact, most biological molecules, right, don't behave that way. And for good reason, because again, that's not compatible with life, right? If you have the molecules inside of your cells always just kind of like reacting with each other in a way that releases tremendous amounts of loose, you know, energy in the form of heat, you know, that wouldn't work very well, right? So let's think about this again. We said a spontaneous direction thermodynamically would be something like glucose going down to carbon dioxide and water, right, to release 686 kcals per mole of glucose in that process. Right? Previously, I used the example of a marshmallow being burned over flame, right? Because that's a really sort of easy, intuitive example of something burning, right? We always think of something as burning, as flame being involved, right? And we said that's because the flame is so much energy that's hitting the glucose molecules in the marshmallow, right? That all the bonds of the glucose molecules are basically being disrupted because there's so much heat energy, right? They just all vibrate apart. And we said, if you have oxygen available, oxygen is a very greedy electron acceptor, right? Oxygen is your sort of quintessential oxidizing reagent. Right? So if there's oxygen available, then as the electrons in the glucose molecules are kind of like freed up, and in that moment, right, they're trying to find better bonds to form with, then oxygen very happily, right, participates with them. Right? But, that definition of burning something, that's combustion, right? So combustion certainly does not happen inside of our bodies. What happens in our bodies is biochemical, right? So the breakdown of glucose, allowing it to do something like interact with oxygen, right, is facilitated by other molecules, right, doing this. So perhaps a more relevant analogy or example would be something like if you had a beaker full of sugar and then you poured something like a strong acid into it. You guys might have done this in high school. This was one of my favorite high school chemistry kind of demos that my teacher did. You mix it together, and what happens is the sugar starts turning brown because it is oxidizing. Right? The sulfuric acid begins to pull away, right? Pull away the electrons in the glucose, and you start actually generating a lot of water as a byproduct. Right? Then as the sugar is breaking down, it goes from brown, which is the same brown as a nice toasted marshmallow over a campfire. But then what happens is you get a lot of exothermic heat generated, and that actually starts boiling the water and turning it into water vapor. And then this big sort of plume, right, of water vapor comes out. You don't want to breathe it because it has some sulfurous gases in there as well. But importantly, what? Even though this is not burning through combustion, right? The same chemical reaction is taking place that can potentially go all the way down past carbon dioxide and water and go all the way down to just carbon, right? Being broken away from things like the carbon dioxide, right? So that's why eventually you get this big thing of charcoal that's kind of getting puffed up from all the steam coming out of the flask. So again, that is an extreme case, right? Because in our cells, we certainly don't use hydrochloric acid or sulfuric acid to break down our sugars, right? We do it in a much more careful, controlled way, right? A controlled way that ends up liberating or getting the 686 kcals from the sugar that our cells can use, right? But we stop at the level of CO2 and H2O being the waste products, right? And each step of the way, right, there are molecules that take care of making the next step, the next step, the next step, right? So that this glucose molecule is not being blown up all at once, like it would be by the sulfuric acid or by like the flame from the campfire. Instead, the glucose is carefully taken apart. And each step along the way, a little energy is released. Right? Just so you know, the burning or the chemical reaction of glucose in this flask with sulfuric acid, if it goes all the way down to just elemental black carbon, right, you would actually release a lot more than just 686 kcals, right? You would expect it to be the release of even more energy. And in fact, you'd go all the way down to negative 920 kcals per mole liberated by the glucose. But again, right, the way our cells are designed, we just stop at the CO2 and the H2O. Why? Right? Because you're like, well, why didn't we just evolve to get like an extra 300 kcals of energy out of the glucose? Because remember, we evolved in the context of living things on Earth first starting off being photosynthetic, right? Plants and, well, early photosynthetic organisms like photosynthetic bacteria evolved first, right? Only later did heterotrophs, like us, evolve to basically take advantage of the sugars that are made by the plants, right? And what did the plants originally evolve to expect as raw materials? They expected atmospheric carbon dioxide and water, right? So therefore, if in reality we burned the glucose all the way down to just carbon, right, that actually wouldn't be very much use for the other forms of life on Earth that are the other part of this cycle. Okay. So what are enzymes then? If we think about what I just explained, that a glucose molecule inside of our cells, right, is not just blown apart and all the electrons are allowed to just sort of spontaneously reform bonds, because that would be too much uncontrolled release of energy, but instead the glucose is stepwise broken down, right? That implies that you have a series of chemical reactions that have to happen, right? And the issue with that is that that series of chemical reactions, right, if you were allowed to just sort of say, well, we'll just let them happen on their own, that might take way too long, right? Because remember, biological molecules are what we call metastable, meaning if you have a glucose molecule, a C6H12O6, hey, it can stay as a glucose molecule like indefinitely, right? Tens of thousands of years, it'll just be a glucose molecule, right? You can't just expect to wait around. Oh, look, one of the carbon bonds broke. Good, right? No, right? Because that's not how life works. You need to be able to accelerate the process of these reactions happening, right? In other words, you have to catalyze reactions, right? A catalyst is something that takes something that naturally would happen and just makes it happen a lot quicker. So if we think about the ball at the top of the hill again, right, the process of pushing the ball to the top of the hill is like energetically the process of plants taking carbon dioxide and water and making something like a glucose molecule, right? You're putting energy into this glucose molecule by making it. So that's a positive delta G. Once you go ahead and shove the ball over the hill, you are releasing that potential energy, right? And that would be a negative delta G. Okay. So enzymes then are catalysts that allow this process to happen at a pace that is compatible for our cells to be able to use them. So why do negative delta G reactions, why do they not typically happen on their own using the types of molecules that we have in our cells, right? Because, right, we need these molecules to be metastable. We cannot just have things like our DNA or our proteins or our lipids just spontaneously falling apart all the time, right? Because how does that work, right? You can't have that. They need to stay together. So that implies that in order to break things down, you need to somehow accelerate the process of certain bonds breaking to provide the ability to liberate energy. Right? So enzymes help this. Right? And how is enzyme activity controlled by our cells? That's what we're going to look at in the next few slides. When you're talking about taking a biologically relevant molecule, right, and having it interact, breaking bonds and allowing it to interact with some other molecule, you have to overcome its kind of metastable tendency, right? Just like the ball sitting on top of the hill, hey, we all know it wants to be at the bottom of the hill because of gravity, but at the same time, it can sit at the top of the hill for tens of thousands of years, right? You need to overcome, it's in the case of the ball, it's inertia, right? In the case of chemical bonds, you have to overcome the fact that the bonds are in fact stable. So let's look at, for example, ATP. ATP, right, we said is an important energy molecule in the cell. So it has this third phosphate group that represents a high energy bond that if you break it, right, the electrons want to form bonds with something else, and the amount of energy contained in that third phosphate bond is pretty high, right? So, but in order to liberate it, though, you actually have to overcome the stability of the ATP molecule, right? Just like we said before with the example of the water molecules forming ice, right? Those weak hydrogen bonding interactions of the ice molecule are easily overcome when you give it enough energy and then they break apart, right? But in the case of these types of interactions, like the phosphate-phosphate bonds in an ATP molecule, these are actually covalent bonds, right? So it takes a bit more energy, but the same principle applies. You got to put in enough energy to actually break the bond, shake the electrons loose, if you want to think about it that way, right? So that amount of energy that you put into it to get that thing to break is what we call the transition state of the molecule, right? And you can see once you put in some extra energy to break the bond, then things sort of take place on their own, right? Because then you'll have the liberation of that phosphate group, and then the energy that's released in the process means you end up with a lower G of the system than what you started from, right? So going from ATP interacting with water, if you convert it down to ADP, so two phosphates in a freed phosphate group, you liberate, right? There's a negative delta G of 7.3 kcals per mole of ATP molecules. So this transition state is really important. One way to think about it is kind of like a light switch, right? You have to put energy into something in order to overcome this barrier of activation, or the E sub A, right? So, so here's a light switch, right? I think we all know what a light switch is, right? It's not like any of you are like, wow, what is that? Right? This is like really advanced cellular biology stuff. So we all know that a light switch, right, has two states. In this case, up maybe is on, down maybe is off, right? But we're all familiar with the fact that when you push a light switch up, it doesn't just fall back down by itself, right? Because that would be useless. What good would that be, right? In order to keep the lights on, you actually have to hold the switch up? No, right? So there's actually physically in the light switch a mechanism, right? Sort of like a hump, like that you actually have to overcome. But once you overcome that hump, what happens? The switch on its own flips, right, down to the lower position. It stays there, right? You can actually hear the sound. It's very satisfying, right? Okay, so that is the principle here, because structurally, right, there is this energy of activation, the amount of energy you have to put into it in order to get it over this hump because of its physical structure, the way the light switch is designed. But once you get over the hump, it will then spontaneously go to this other state, right? So think about this. In a complex molecule, right, there is this idea of physical structure, right? The angles of all these bonds, right, actually form the metastable nature of the molecule. And if you really want to tweak or break something in that bond, you have to overcome one state of the molecule so that it can then, once you get over that hump, right, it'll sort of naturally assume another state where it's more comfortable again, energetically. One thing to think about is one way that you could make this easier is, let's say I'm really lazy, right? Oh, it takes more energy to push this thing over the hump than I want to spend, right? It takes too long for that to happen. You can make it easier, right? By doing something like, well, here I have a rock tied to a string, right? Yeah, I know this is this whole like state university budget thing, but anyhow, right? So if I were to tie a rock around this light switch, can't even do that. Right? Now you have this rock pulling down on the light switch. Suddenly what? It makes it way easier for me to flip it, right? Because the rock is helping out. Importantly, it does not change the amount of energy represented by this switch when the switch is up versus down. It just makes it a lot easier to get over the transition, that hump, by having a rock tied to it. Right? Okay. So catalysts then lower this energy of activation, right? Just like the rock hanging on the switch makes it a lot easier for me. I have to put in less energy with my finger in order to get the switch to move, right? A biological catalyst does the same thing. It lowers the energy requirement of the transition state. Therefore, right, a particular reaction will happen much easier and therefore much faster than if left by itself. So here, what we're talking about is this blue line would represent the transition state of this reaction of ATP going down to ADP if it is catalyzed by something, if you have a rock hanging on the light switch, right? So all molecular catalysts increase the reaction rate by lowering the required energy of activation to make the, bond, breaking of bonds, reforming of bonds happen. They work by forming temporary interactions with targets that sort of stabilize the transition state, making it easier to overcome. And importantly, just like I said with the light switch, just because you have a catalyst helping out, it does not change the starting energy or the final energy, right? The ATP going to ADP is still going to be the same drop in energy, the liberation of minus 7.3 kcals per mole, whether or not that reaction is catalyzed. Right? So what are these molecular catalysts, right? These are molecules that function inside of our cells, right? Our cells make these and they help to accelerate different important biological processes. So let's get a little bit closer to home with something that actually happens inside of our cell. Here is hydrogen peroxide, H2O2, right? Peroxide is a very strong oxidizing reagent. That's why, if you use peroxide, you can use it as a bleaching compound, lightening hair color, things like that. Importantly, peroxide is also used as a first aid application for killing germs like bacteria. Why? Right? Because peroxide is really greedy in terms of grabbing electrons, right? That's because of the O2 part of the H2O2. Remember, oxygen loves to grab electrons and oxidize things. So if you have things like proteins or other biological molecules and you expose them to peroxide, right? If the oxygens are taking the electrons away from the normal structure of these molecules, things can happen from, for example, the pigments in your hair that are a function of different proteins, right, being altered or destroyed that way. So therefore you end up bleaching your hair, right? Or the cell walls of things like bacteria, right? The peroxide is basically destroying the cell walls of the bacteria by stealing all the electrons away so that the, molecules that make up the integrity of the cell wall of the particular bacteria is disrupted, and the bacteria then lyse and blow up, right? Peroxide is a normal waste product that's generated by the living functions of our cells, like the metabolism in our cells. Right? So you can imagine then that if peroxide is building up inside of our cells, that can cause a lot of damage, right? It could start actually destroying or disrupting normal proteins inside of our cells. So our cells need to get rid of it, right? Our cells need to get rid of peroxide as it's building up. The problem is peroxide by itself, if you just sit around and wait for peroxide to on its own break down into H2O and O2, right? Because H2O and O2, hey, that's nice, right? Because you're not going to get hurt by H2O or O2. So let's just wait around for the peroxide to do that. No, you can't do that, right? Because it takes many hours for H2O2 to just break down by itself. And our cells cannot afford that, right? So we need to catalyze that reaction to neutralize the peroxide. Iron ions, Fe3 plus ions, can actually serve as a very strong molecular catalyst to break down peroxide. Over here, you can see the structure. So here's peroxide, right? And it's funny, you never think about it, but peroxide is super simple, right? It literally is 2 oxygens, 2 hydrogens, okay? You can see if there are iron ions present, the iron will interact with the oxygens. Right? And by doing so, what does it do? It disrupts the oxygen-oxygen bond here in the peroxide, right? Because then the oxygen might really want to then, interact with the iron more. And you can see the iron ion kind of is causing a problem here, right? It's disrupting the structure of the peroxide. And by disrupting it, it makes that oxygen-oxygen bond a lot easier to break. Okay. In fact, it's so effective that if you just have some iron mixed in here with the peroxide, it increases the rate of breakdown of the peroxide by 30,000 times faster than if you just left the peroxide by itself. Okay. So what does this have to do with how our cells deal with peroxide? Guess what? Our cells use iron to break down the metabolic waste products like peroxide that are building up in them. We have so our cells cannot afford to wait, right, for the peroxide just to break down by themselves. And you also cannot just rely upon having, hey, great, let's just have some iron floating around in our cells, right? So that hopefully the iron will bump into the peroxide. Because again, right, you have no control over that, right? If things are just mixed together and you wait for things to hopefully come together, biochemical reactions, living biochemistry is all about having certain molecules facilitating other molecules coming together at the right place and the right time, right? So we have a protein known as the catalase enzyme. And what that catalase enzyme does is represented here by the shape. It's the right shape to bind to peroxide, right? And by binding to peroxide, what it does is it actually accelerates the ability of iron to come in contact with it. Why? Here is the structure of the catalase enzyme. So you can see it's a quaternary structure. It's made up of different units of the individual catalase proteins. And these units have a binding site for iron. So in other words, the catalase enzyme, this protein, is carrying around the iron, and it's also the right shape to bind peroxide, thereby bringing the iron and the peroxide together much more rapidly than if you just were waiting for peroxide and iron to float around and randomly bump into each other, right? And so by bringing these two things together, it then facilitates the rapid dissociation or breakdown of peroxide, right? So because we have this catalase enzyme in our cells, right, it increases the rate of the peroxide in our cells being broken down about 100,000 times faster than if the peroxide was just left alone. So not surprisingly then, most enzymes are proteins, right? Because we often think about proteins as being complex three-dimensional mini machines, right? We learned that proteins, if we think about them as being a polypeptide chain of amino acids, right? You have the carboxyl terminal end of this polypeptide chain, right? The N-terminal end that has the free amine group from this very first amino acid residue here. And then you have follow this chain, right? Here's amino acid number 10, number 20, number 30, right? 40, 50, et cetera, eventually you get to the end. And this chain of amino acid residues is 129, right? So this protein is 129 amino acids long. And then the very last amino acid we call the carboxyl end of the chain, right? Because that amino acid has its free carboxyl group sticking out the other side. Along the way, you might have certain amino acid residues like this glutamate at position 35, tryptophan at 63, alanine at 107, right? And you might say, well, these are really far apart from each other, right? But they're only far apart from each other if you're thinking about this as a linear structure, right? Then to say 35 to 63 to 107, right? Those are far apart from each other. But remember, the actual protein itself, when properly formed, folds up, right? And here it is folded up. All 129 amino acid residues folded up in the normal intended tertiary structure of this protein. And look what happens, right? Those amino acid residues, the glutamate 35, alanine at 107, tryptophan 63, aspartate at 52, suddenly are actually close to each other, right? And these form the active site of the enzyme. The identity of those amino acids, right, are specifically intended because the shape of their side chains contributes to the shape of this active site. And that's where whatever molecule this enzyme is going to work on binds to. So whatever the target molecule is that this enzyme is going to work on, we call that the substrate, right? That's where it's going to bind in this active site, right? That's where there's going to be activation of the enzyme activity, and that's where catalysis, right, or accelerating the process of an intended molecular interaction takes place. Right. So here's another way to appreciate it. Right. Here, let's say we have an enzyme that works on a particular substrate. So imagine, just hypothetically, that this substrate, which is some sort of, relatively simple molecule, it's made up of these, orange, I don't know what these are, maybe these are carbon atoms or something like that. And then there's a blue dot on it representing the fact that it has an oxygen group on it. And let's imagine that our cell, in order to use this molecule, needs to take the oxygen off. Right? So it needs to be deoxygenated. So the little blue oxygen needs to come off. Right? Again, you could wait a long time for the oxygen just to fall off by itself. But this molecule, it's metastable, right? Meaning who knows how long it takes for the oxygen to fall off by itself. Might be hours, might be centuries, right? Might be 10,000 years. So your cell's not going to wait around that long. So your cell has enzymes that can catalyze this reaction. So look at the fit. Here's the protein. Here would be the active site, right? In the active site on the left here, you can see here is the substrate with its oxygen attached to it. These purple things are the side chains of the amino acids that are part of the protein. That when the protein is folded up in this way, these side chains are all part of this active site. And look how they create the right shape to be able to bind this particular substrate shape in it, right? How specific this enzyme is for this particular substrate. So the enzyme, which is capital E, binds the substrate, capital S, right? And you have this temporary state where you have an ES, right, where they are together, and now you're activating sort of the function of the enzyme, and the enzyme is then going to catalyze the reaction, releasing some sort of product. We can look at this in an animation here. Right? Look at how the substrate with the oxygen, right, starts off in an oxidized state when it fits into the pocket, but then, right, as the pocket changes shape, what is it doing? Look at the substrate itself. It's getting torqued, right? It's getting shoved around or bent in such a way, right, that it suddenly makes many of those bonds unstable. And in particular, the bond between the oxygen and the rest of the substrate molecule. The reshaping of this binding site of this protein, right, involves almost the entire protein shifting around. You can see it kind of moving, right? And you can almost imagine, right, that if this was like a physical thing that you could hold in your hands, right, and you kind of like twisted it, right, something could happen to something that's in there. So we have many sorts of household devices that are kind of like that, right? Where you kind of like have something in order to pop something out, you put it in there and you twist it and something can like come out, right? So how does this twisting, what does this have to do with the protein? It has to do with the fact that the tertiary shape of the protein is being altered in such a way, right? So that changing of shape gets kind of transferred to the changing of shape of the substrate, causing this reaction to happen very quickly and the oxygen popping off. Okay. Why are enzymes then very sensitive to temperature and pH? Well, hopefully you can see, because it all depends on the protein being the exact right shape, tertiary structure that it's supposed to be in order for it to work, right? If it's denatured due to temperature being too hot, right, or the pH being too acidic, then the enzyme is not going to work. So that goes all the way back to our discussion of using things like Taq polymerases in doing PCR, right? Because human polymerases, DNA polymerases, evolved to be the right shape at 37 degrees Celsius, right? They won't work at 80 degrees Celsius, right? Because their shape is not going to be right anymore in order for them to do their enzymatic function. But something like Thermophilus aquaticus bacteria evolved to have, their version of DNA polymerase be the right shape at like 80 or 90 degrees, right? So therefore, at that temperature, their polymerase enzyme works properly. So this is what we're talking about then, right? You can think about it as an enzyme is essentially lowering the amount of energy required then for something like a bond to break, right? And that can happen by distorting the shape of the substrate molecule, which is what we just looked at in the previous example, right? Or enzymes might donate or accept protons to change the shape of a substrate, or enzymes may accept or donate electrons, right? Maybe forming a bond temporarily with the substrate as part of changing the shape and facilitating the substrate to undergo the desired reaction. Okay. So enzymes then can be regulated. So on that animation where I showed that the whole enzyme is changing its shape, what that implies is that if you have something that comes along and binds to that enzyme, and changes its shape, that might be a good way to actually make the enzyme do its function. So this has to do with allostery. Allostery means if you have a molecule and something binds and changes the shape in one region, that there is that kind of ripple effect, right? And then the whole molecule, like the whole protein, might have to change its shape a little bit to adjust for it, right? It's just like the light switch. If the enzyme starts in an off position, that means energetically it's very happy in this particular conformation, right? But if something comes along and pushes up against it, it kind of gets it to kind of change its shape, and then it'll assume the next sort of comfortable shape, right? And in the process of doing so, that might be tweaking the little substrate in the enzymatic pocket, causing the desired molecular reaction to happen. So here's an enzyme. Here we have a site on the enzyme where an allosteric activator, this green thing, might come and bind to it, and that causes this ripple effect, and the enzyme changes its shape. As a result, it then maybe opens up some other surface on the enzyme where a substrate can now bind, right? And therefore, the function of this enzyme is controlled in an on and off kind of way, allosterically, by this other allosteric activator being added to it. One of the most common ways that this is done is if that thing that's added to it is a phosphate group, right? So phosphorylation of proteins post-translationally, right, is a very important and common way that certain enzymes are switched on or switched off. Okay. All right, so we're over time today. This last slide with this stapler is an analogy that I often use to think about how enzymes function and get you thinking about the evolution of enzymes. All right, so I will save this for the start of next lecture, where we're going to explore this concept of how enzymes evolved and how the catalytic sites of enzymes are oftentimes highly conserved through evolution. Okay, all right, so I'm going to go ahead and stop there.